DMLT Second Year Microbiology Supplementary 2021. Question number 1. Classify and describe the hypersensitivity reaction. There are 4 types of hypersensitivity reactions as below. Type 1 reaction mediated by IgE antibodies. Type 2 cytotoxin reactions mediated by IgG or IgM antibodies. Type 3 reaction mediated by immunocomplexes. Type 4 this is delayed reactions mediated by cellular response. Now, type 1 hypersensitivity is also known as immediate reaction and involves immunoglobulin E mediated release of antibodies against the soluble antigen. This results in cell deglutation and release of histamine and other inflammatory mediators. Type 2 hypersensitivity reactions refers to an antibodies mediated immune reaction in which antibodies means IgG or IgM are directly against cellular or extracellular matrix antigens, resulting in cellular destruction, functional loss, or tissue damage. Type 3 hypersensitivity is designated as immunocomplex hypersensitivity. This reaction occurs through the formation of antigen antibody complexes that activate complement and result in tissue damage. The fourth type is considered as a delayed hypersensitivity reactions because it usually occurs more than 12 hours after exposure to the maximal. Question number 2. Define sterilization. Write in detail about chemical sterilization. Sterilization means describe a process that destroy or eliminate of all microorganisms, all forms of microbial life, and is carried out in healthcare facilities by physical or chemical method. Chemical sterilization involves the use of chemical agents to kill unwanted microorganisms such as fungi, bacteria, virus, and other disease-causing agents. Liquid and gas sterilizations are chemical method of eliminating all viable microorganisms and spores. What are the advantages of chemical sterilization? The advantages of chemical sterilization are that it is relatively inexpensive and fast acting, efficient. Most often, it does not require expensive and complex equipments. Then, what is an example of chemical sterilization? One example of chemical sterilization is that use of ozone to decontaminate medical devices means elimination of infectious agents from medical surface and equipments in to protect healthcare provider. What is the chemical method of sterilization? Sterilization eliminates bacteria, virus, parasites, fungi, and spores. Chemical sterilization involves the uses of chemical agents to kill these unwanted microorganisms. Question number 3. Write in brief about structure of bacterial cell. Bacteria are the small single cell organisms. Bacteria are found almost everywhere on the earth. In bacteria, the cell walls form a rigid structure of uniform thickness around the cell and is responsible for the characteristics shape of the cell. For example, if it is rod shape, convex, spiral, etc. Inside the cell wall means rigid layer in the plasma and cytoplasmic membrane. This is usually closely opposed to the wall layer. Now, structure of cell. A cell consists of three parts: the cell membrane, the nucleus, and between the two, the cytoplasmic within the cytoplasma. These agreements of the fine fibers and within the cytoplasma lies integrate agreement of fine fibers or even thousands of minuscule but distinct structure is called as organelle. Here diagram is also there, bacterial cell wall. Then you have to draw the diagram and label it. Organized into a three categories. Bacterial cell is organized in three categories: internal structure, cell involves, and external structure. Internal structure means cytoplasma. Then you have to go through this. Question number 4. Mode of transmission included contact transmission, direct and indirect. Person to person transmission is the form of direct contact transmission. Here the agent is transmitted by physical contact between two individuals through actions such as touching, kissing, sexual intercourse, or droplet infection. Means that is a cough, coughing, seasoning, touching, and all body fluids like that. The term mode of transmission refers to how an infection agent, also called a pathogens, can be transmitted from one person or object or animals or to another form. Those are virus, bacteria, parasite, and fungi. Can be spread infectious diseases. Then these direct and direct contact means infectious disease are often spread through direct contact types, that is including person to person contact. Already I have told person to person how it is contact means. Already I have told that is here chain of infection is there. You can observe the image and you can understand. Then droplet infection means through wind, through cough, through seasoning. These are the infectious disease will spread from one person to another person. Germs get around us, means when contact hands, toys, and when shaking the hands, this is germs get around us, which are the germs are there around us. That is how germs will be get, that is the portal entry, means how they will enter into in our body, means mouth, eyes, and cutted in the skin, like that. Okay, then agents, which are the agents, means bacteria, virus, and parasites. Where they will reservoir, those are reservoir in people's, animals, pets, dogs, cats, and wild animals also. Sometimes which we are eating in the food, and soil, means when you are working in the soil, in the garden, and contaminated water. These are the reservoir of infection. When these are the indirect contact infectious. Okay, then susceptible host, means who will get the infection, means small babies. When you are touching from one baby to another baby in the hospital or in the household level, mother has to maintain the hand hygiene. Okay, like that. Children and elderly, means old age people, then who are having very less immunity power, those person. Okay, then who has not immunized children, that is also. These are the susceptible host. Those are come under the risk factors. This is also come under the mode of transmission. Question number 5. Uses of compound microscopes and care. 5 uses of compound microscope. The compound microscope is commonly used in forensic science laboratories to observe the trace evidence such as hair, fibers, biological fluids, pathogens, grains, etc. The compound microscope has various applications in biology, pathology, physics, forensic, and other fields to examine of the samples and analysis of the tissue. In research field, especially in biological research, one of the first way as to how compound microscope is used in the field of biology. Not only does it help you see the microorganism, but you can also see the bacteria, cells, as well as tissue very easily. They are widely used for research, not only in biology, but also in the microbiology and genetics also. The compound microscope is of a great use in pathology lab, so as to identify the disease and diagnostic purpose. In the forensic laboratory, compound light microscopes are used to identify presence of minerals, metals in human cells, so as to solve the chemical causes. In forensic medicine, expert can also find out the origin of a drug by viewing its component and particles under the microscope. These are the uses of microscope. The microscope. Before and after doing procedure, wash your hands and sanitize before touching of the microscope, after touching of the microscope, as well as when you are taking the food also. Always wash with soap and water if your hands are visibly solid. Okay, try to minimize touching of your mouth or nose with other our hands also. Avoid sick people if possible. Wear disposable gloves to avoid contact with blood and feces. Always you have to performing the procedure all protective barriers, mask, gloves, everything you can use it. Use the disposable gloves when caring for an ill person. Cover your mouth when you are seasoning and cough and wash your hands. This type of care, and after completing the procedure you have to wipe it microscope properly and cover it and keep it in the laboratory. Number 7, reagent use in gram staining technique. Reagents needed for gram staining including crystal violet, that is a primary stain. Number 1, grams iodine solution, acetone, ethylone, and basic solutions are also. Reagents used in the gram stain. The gram stains are applied to apply of bacteria on microscope slide in the following order, that is crystal violet, grams iodine, and recoalizing agent and saffron. The gram stain uses in 4 stains. Reagent, that is as I have told, crystal violet, grams iodine, ethylene, and saffron, and crystal violet. These are the primary stains. Enter of the bacteria, giving them a purple color. Gram technique means gram stain involves applying a stain to a sample in glass microscope slide and looking at it under the microscope to determine if bacteria are present at all. Gram staining is a common technique used to differentiate two large group of bacteria used on their different cell wall and constituents. Gram stain means is a different staining procedure used to bacteria gram stain and gram negative and gram positive based on the chemical and physical properties of their cell walls. The bacteria are differentiated through the series of staining and decolorization steps. Gram positive cells will stain purple and gram negative cell will stain red to pink. And supply and reagents, there are different. Okay, that is a personal protective equipments, slide rack, and absorbent paper, and water, and crystal violet, grams iodine, and decolorizer, saffron. These are the, and etc. Then pencil. These are the all supply when you are doing the gram stain. Okay, then some of the instructions are there, and gram staining procedures that steps is also involving. Okay, then this is the procedure. Place the prepared fixed smear on a slide rack. Then flood the slide with crystal violet. Then wait for 15 seconds. Then rinse the slide with water. Then third one, flood the slides with grams iodine. After 15 minutes, rinse the slide with water. Apply the decolorizer to the slide and rinse the slide immediately with water and flood the slide with concentration. Then wait for 15 seconds. Then rinse the slide with water and blot the slide with absorbent paper. Be careful not to wipe the cell of the slide. Okay. Next, allow the newly stained slide and dry completely. View the slide under oil using the oil and objectives for a total manifestation of then what is the range is there according to the lab, that also you have to observe. And last, then record the results, means that is the interpretation. Gram staining procedures, these are the instruction when you are performing. Step 1 is the crystal violet, step 2 is the iodine, and step 3 is the alcohol, step 4 is the saffron, which you are observing in the slide. Views is also there, you have to observe that. Then crystal violet means what is there, that is under writing is there, you have to go through that. Question number 8, contribution of Louis Pasteur. He was the first to demonstrate the infectious disease are caused by the microbes and disproved and concept of spontaneous generation, means that is the idea that microbes could appear out of nothing also. Okay, then develop the procedure of pasteurization. He was developed the procedure of pasteurization as well as being it and being in presence of his observation, and he has developed the some of the world first vaccination, and he was studied the molecular and discover the microorganism cause for the disease, and originated the process of pasteurization, saved and beer, wine and silk industries in develop the and also develop the vaccines against anthrax and rabies, means rabipur. Then ARV ARV vaccination he has identified, which has given for dog bite patient. Then again he has done one cell theory. He achieved it by revising the experiment that demonstrated that cell would only grow and they were exposed to air. That is called as cell theory. Then vaccination has production also he has started, then which he has identified the cholera, anthrax, rabies, and September 28th 1895 he has implementation in the vaccine. Okay, Pasteur made a virtually one of a kind contribution to science. Then you have to proud of him. Okay, technology, medications, and he has established the pasteurization procedure, then which has done in the milk, means in Hubli Dharwad, in between, that is milk pasteurization is going on, because if without pasteurization means person will get the disease. So he has identified that procedure. And even though then wine also, beer, silk industries also, then which are harmful for human being, that also he has identified. And means you have to be take precautions yourself, that also. Then he was studied in molecular also. Okay, then you have to proud of him. This much is in question number 9. Innate immunity. Innate immunity is the first response of the body's immune system to a harmful foreign substances. When foreign substances such as bacteria, virus enter into our body, certain cells in the immune system quickly, means as early as possible, response and try to destroy them. That is the first innate immunity. In innate immunity, there are types. They protect the non-infected cells from infection. Immunity presents by birth is called as innate immunity. It is 4 types: physical, physiological, cellular, and cytokine. This type of immunity is not as strong as acquired immunity. Means when that is which you have learned in the class, that is natural immunity and acquired immunity. Natural immunity means nothing but innate immunity, which in the birth which you will get. Okay, then property of innate immunity. Innate immunity is comprises of different components, including physical barrier. Means physical barriers means when you are touching, some of the physical reasons is also there, some of the pathological reasons is also there. In our body, mucous membrane sometimes, that is a pathological reason. Epithelial cell tissues, that is also our pathological reason. Some of the physiological means when you are touching the in outside atmosphere. Automatic barriers means those are epithelial and phagocyte cell enzymes. Okay, and inflammation. When you will get the inflammation means that is the white blood cells in our body, and some of the signs and symptoms also we will develop. Okay, then now advantages of innate immunity. Innate defense are mobilized quickly and provide a critical line of defense against pathogens, and responses do not change with sequential exposures. Okay, components of innate immunity. What are the components means? That is the physical barrier, already you know that. That is the cellular components and humoral responses. Humoral responses means when foreign body will enter in our body, that innate immunity will suddenly react and suddenly which are the foreign substances are there, immediately that will fight against and destroy them. Okay, these are the component. Question number 10, bacterial growth curve. The bacterial growth curve represents the number of live cells in a bacterial population over a period of time. There are 4 distinct phases of the growth curve. Okay, then that is the initial phase is the log phase, where the bacteria are metabolically active but not dividing. There are 4 phases of bacterial growth curve. Lag phase means no increase in number of living bacterial cells. Number 2, log phase means exceptional increase in number of living bacterial cells. Third phase, stationary phase means in a number of living bacterial cells, rate of cells division and death roughly equal. Fourth phase, death or decline phase, and exceptional decrease in number of living bacterial cells. Important points of the bacterial growth curve. The bacterial growth curve represent the number of live cells, as I have told in 4 phases how that changes will take place in the bacterial population over a period of time. There are the 4 distinct phases. Already you know that 4 phases. This change will be takes place in all systems. Means question number 11 only 3 marks. Means a structure of antibody. Here picture is also there, then you can draw the diagram and you have to write it a little bit. An antibody has a Y-shaped structure made up of 4 polypeptide. Each subunit has 2 identical light and heavy chains. The N termines of each heavy chain form an antigen and binding domain with the light chain. There are 2 antigen binding domains forming the arm of the Y shape. Type of antibodies, each with different functions. There are 4 types of heavy chain constant regions in antibodies, that is also called as immunoglobulin. And according to these types, there are classification into IgG, IgM, IgA, IgD and IgE. There are the then here picture is also there. You can draw the diagram, which are the heavy chain, light chain, then everything is displaying in this. Question number 12. Write a neat diagram of bacterial spore. Bacterial spore is a small whole structure that is highly resistant to environmental factors like heat, radiation, desiccation and chemicals. Bacterial spores are the small whole and resistant to high temperature, radiation and chemical agents. Already I have told that. When they are formed intellectually, they are called as endospores. The bacterial cell producing spore is called vegetative cell. Here diagram is also there. You have to draw the diagram. Then functions. Bacterial spore are the several mechanism that ensure the bacteria are several. They can remain viable from millions of years. Okay, then when the bacterial spores, gram-positive bacteria, are known for producing the endospores, which are observing, means endospores. Just now I have told that is a intellectual. Then they are intellectual. They are called as endospores. And when they will endospores are producing, that is a question number 13. Cold sterilization. It is a process in which sterilization is carried out at low temperature with the help of chemicals, filtration, radiation, and all other means excluding high temperature. It is done for products that contain heat sensitive integrates and required sterilization. Cold sterilization is the use of liquid chemical solutions to sterilize the medical instruments. Radiation is referred to as a cold sterilization. That is the key point. Then food processing and by radiation, and that is in pasteurization, cold pasteurization. These are the examples. Okay, in physical non-thermal made up of food preservations, there also they will use the cold sterilization. Radiation cause minimal modification in the flavor, means color, nutrients, taste and other quality attributes of foods. Cold sterilization importance. Cold sterilization provides protection and low temperature molecular stabilization. Question number 14. Name 3 gram-negative cocci. Gram-negative cocci bacteria including clinically important in such Neisseria, gonorrhea, Clostridium tetani, Streptococci, Staphylococci, Salmonella typhi, which are there in display in your image view. It is there, you can read. And here one diagram is also there. You have to draw the gram-negative cocci diagram. Question number 15. Cleaning of body fluid spill in laboratory. When you are working in the laboratory, if it is any fluid spill on your body or clothes, means how you are going to clean. First point, wipe the area with water and detergent until it is visibly clean. Next, saturate the area again with sodium hypochlorite, means 0.5%. This is a 1:10 dilution of 5:25% sodium hypochlorite bleach, which should be prepared daily. Rinse off the brush and pan under the running water and place it in dry place. Drop of blood or other small spill can easily be managed by wiping the area immediately with paper towel, then cleaning with warm water and detergent, followed by rinsing and drying the area. Dry the area as wet areas attract contaminants. The following steps should be taken during spill cleanup. Prevent the spread of dust and vapors. Then neutralize acid and bases. If possible, control the spread of the liquid and or absorb the liquid. Collect and contain the cleanup residues and dispose of waste. Decontaminate the area. Question number 16. Enrich media. Enriched media is a type of culture media which is used to growth microorganism of a wide variety. Enriched media. This enriched media is contain high nutrition in the form of egg yolk or blood serum, etc. Its basal medium also contain nutrients. Enriched media is use the certain growth media to favor the growth of particular microorganism over others. Enriched media contain added essential nutrients specific organism need to growth. Enriched media, this is the prepared by adding additional substances like blood serum or egg yolk in the basal medium. It's used to growth microorganism as they require additional nutrients and growth promoting substances. For example, are chocolate agar, blood agar and Leo Fleischer serum slope. These are the types of enriched media. 17. Define precipitation with example. Precipitation in microbiology, it is a chemical reaction in which you mix two solutions of two ionic substances and solid ionic substances that will form, that is called as precipitation. Next one, another, precipitation is a process where the soluble antigens bind with their specific antibodies at an optimum temperature and pH, resulting in the formation of an insoluble precipitate. The interpretation between the soluble antigen and antibody results in the form of any insoluble tactics that forms a precipitate out of the solution. Examples: rain is a liquid water precipitation and snow is a solid water precipitation. Precipitation is a liquid or solid that falls from the atmosphere to the earth. This is the example of rainwater is the precipitated, solid water is the means snow precipitate. Question number 18. Preparation of saffron stain. Add a 20 mg saffron powder to a 100 ml of beaker. Pour 20 ml of distilled water into the beaker and make 0.1% saffron in staining solution by constant treating. Transfer 20 mg of fast green dye in another 100 ml beaker. Moreover, make it a 0.1% staining solution by adding 20 ml distilled water in it. Saffron also as a basic red to is a biological stain used in histology and cytology. Saffron is used as a counter stain in some staining protocols, coloring all cells nuclei red. It can also be used for the detection of cartilage and mucin and mast cells and granules. Question number 19. Functions of bacterial capsule. The bacterial capsule serve as a shield, giving protection from toxins and from drying out. Capsules allow adhesion to surface and help enable the bacteria to evade the host immune system. The water contained in the capsules gives the protection against drying out. Attach certain pathogenic microbes to host cells in resistant pathocytosis. Enhance the organism's ability to cause disease. Bacterial capsule not only promotes bacterial biofilm formation but also help bacteria evade immune host system. And capsule will take a very important role to determine the bacterial infection and to prevent efficient barrier for bacteria killing by environmental threat. Question number 20. Name gases, disinfectants. 4 gases are there: formaldehyde, carbon monoxide, methylene bromide, ethylene oxide. To disinfect, clean surface and the air. They can be used to eliminate bacteria, virus and other microbes, which are frequently composed of chlorine, ozone and hydrogen peroxide. Gases disinfectants. Formaldehyde gas used to sterilize air in rooms exposed to continue for 3 hours. Spray ammonia to remove formaldehyde residues. Ethylene oxide kill microorganisms and spores. Used for plastic and rubber articles. Question number 21. Fumigation. Fumigation is a method of pest control or the removal of harmful microorganisms by completely filling an area with gases, pesticides or fumigants to or poison the pests within. Fumigation is a powerful contamination method within microbiology laboratories, effective in eliminating microbial growth. Types of fumigation. Fumigation is a method of eliminating pests and insects from houses, buildings, and proceed accommodates by using various chemicals. One is gas fumigation, solid fumigation, liquid fumigation. These are the three types of fumigation. Question number 22, bacterial loops. An inoculation loop, also called as a smear loops. Inoculations wand are micro stickers. It is a simple tool used mainly by microbiologists to pick up and transfer a small sample of microorganisms called inoculum from microbial culture, that is for sticking on a culture plate. This process is called as inoculation. The tool consists of a thin handle with a loop about 5 mm wide or smaller at the end. It was originally made of twisted metal wire, but disposal molded plastic versions are now very common. The size of the loop determined the volume of liquid an inoculation loop can transfer. An early report of the use of an inoculation loop as an analytical tool was by O. Sullivan. Question number 23, different types of infection. An infection occurs when a microorganism such as bacteria, fungi, virus enters a person's body and cause harm. The effects of infection occurs due to the immune system's attempt to get rid of the invading organisms. Types of infection: primary infection, reinfection, secondary infection, hospital acquired infection or nosocomial infection, cross infection, subclinical infection, latent infection, focal infection. These are the various types of infection. Question number 24. Name 3 different active and passive immunity. Active immunity refers to immunity which results from the production of antibodies by the person's own immune system in response to a direct contact of an antigen. In passive immunity refers to a short-term immunity which results from the introduction of antibodies from the outside. Active immunity mediated by the antibodies produced by the person's own cells. Passive immunity mediated by the antibodies produced outside the body. Active immunity, the pathogens has direct contact with the body. In passive immunity, pathogens does not have direct contact with the body. Active immunity does not generate a rapid response. Passive immunity generates a rapid response. Active immunity may last for a long time. Passive immunity may not last for a longer time. Question number 25. Bacterial filters. Through the respiratory filters, also known as bacterial or microbial filters, are devices with a high capacity to prevent the passage of microorganisms and are placed in the breathing circulate in order to protect the patient from possible respiratory infections carried by the respiratory. Filters can have a different mechanism of microbial filtration. Number one, mechanical filtration, electrostatic filtration, and filtration bactericides. These are the bacterial filters. Question number 26. Name 3 live vaccines. A live virus vaccines helps the body's immune system organize and fight infections caused by the non-weakened from the virus. Example, live virus vaccines are the chickenpox vaccine and the measles, mumps, and rubella. That is called as MMR vaccines. Live bacterial vaccines, immunococcal and pneumococcal vaccines, diphtheria, tetanus, whooping cough, polio, measles, mumps, and rubella, MMR, chickenpox, varicella, influenza, flu. These are the various live vaccines are there. Number 29. Name 3 transport media. Buffered glycerol, saline medium, liquid bacterial transport medium, ballard medium, semi-solid bacterial transport medium, chlamydia transport medium, liquid bacterial and viral transport medium. Transport medium is a buffer solution that contains nutrients and other components to preserve the viability of bacteria during transport without allowing them to multiply. The main purpose is to keep the sample as close to its original state as much as possible. Transport media can be classified by physical state, solid, semi-solid, and liquid, and by utility, bacterial, viral, parasite, transport media. Here are the some examples are there transport media. Question number 30. Gram-positive bacteria cell wall. Gram-positive bacteria have a thick cell wall that contain a large amount of peptidoglycan, a polymer that's 20 to 80 nanometer thick. The cell wall also contains other components including lactic acid, polysaccharide, and protein. Peptidoglycan, A thick surrounds the cytoplasmic membrane. Ticlonic acid localized in the cell wall along with lipotactic acid. Polysaccharides decorate the peptoglycan. Protein decorate the peptoglycan. Here diagram is there. You can see the diagram and draw it neatly.